Hello, everyone, and welcome to today's webinar. I'm Young Lee, the President and CEO of the Damon Runyon Cancer Research Foundation. Today, I'm delighted to welcome you and thank you all for joining us. Today is our latest in our Damon Runyon Cancer Research Foundation Catalyst webinar series featuring Damon Runyon scientists. Damon Runyon's mission is simple: funding brave and bold. We provide funding to the most brilliant young scientists to enable them to pursue high-risk, high-reward research that leads to breakthroughs in the understanding and treatment of all cancers. We have a long track record of success, and over the past 75 years, we have invested over $430 million in nearly 4,000 scientists. Today, we'll be talking about and focusing on what the next big breakthrough in cancer research is. I'm delighted to turn over the program to our moderator, Julie Segre, PhD, who is a former Damon Runyon Fellow, as well as a current member of our Innovation Award Committee. And she's a senior investigator at the National Human Genome Research Institute, National Institutes of Health in Bethesda. Her lab focuses on a field called microbial genomics, which she will explain to you more in just a moment. And she will introduce our distinguished panelists. After each panelist presents, we will have a discussion and Q&A session moderated by Dr. Segre. You can type your questions in the Q&A box in the chat, and we will ask as many questions as time allows. This webinar will conclude no later than 5 p.m. Eastern or 2 p.m. Pacific. Julie?
Wonderful. Thank you, Dr. Lee. I'm Julie Segre. Thank you for that nice introduction. As Young mentioned, I was a Damon Runyon Fellow 20 years ago at the University of Chicago, and I can tell you that the support and investment in my career from Damon Runyon has been invaluable. I am a leader in the Human Microbiome Project, understanding how the bacteria, fungi, and viruses that live in and on our bodies promote health and are altered in the context of disease. I've also developed the field of real-time microbial genomics to track antibiotic-resistant bacteria in healthcare settings, break transmissions, and protect some of our most vulnerable transplant or immune-suppressed cancer patients. To sort of put this webinar in context, 5 years ago, I was speaking with an uncle, a gastroenterologist who had just been diagnosed with malignant melanoma, and he'd provided care for a lot of colorectal cancer patients in his 30 years of practice. He and I were talking, and he told me, I used to tell some patients that new treatments were right around the corner, so maybe they should consider that when making treatment decisions. And I would say that, and sometimes I was just afraid that they couldn't tolerate or benefit from the most aggressive treatments that they might hear about. But now it's really true. And he's telling me this as both a gastroenterologist and as a cancer patient. He said, there are so many exciting new treatments for cancer. And it was speaking of the breakthroughs in understanding how our immune system can help us fight cancer and how the microbes that colonize and infect us regulate this immune system. So we're joined today by early career clinician scientists who are leading these breakthroughs. And it's an honor today to be joined by Dr. Kyle Daniels from the University of California, San Francisco, a former Damon Runyon Fellow with expertise in immunotherapy and a background in biophysics. Allison Didychuk from Yale, also a former Damon Runyon Fellow and current Dale Fry scientist with expertise in molecular virology and structural biology. And Dr. Rabi Upadhyay from New York University School of Medicine, a current Damon Runyon physician scientist with expertise in cancer immunotherapy and the human microbiome. So we're going to start with brief presentations about their individual research programs and scientific careers, and then move into a Q&A discussion. So we're going to start with Kyle Daniels, and I'm going to let you introduce something about your very exciting work. So thank you. Oh, Kyle, I think you're still muted.
Great. Can you hear me now? Okay. So thanks for the introduction and getting the conversation started. So my work, which is Damon Runyon funded, and was enabled by the generous support of the foundation, is based on using a very simple idea to fight cancer. So in my work, I'm trying to harness cell signaling to control immune cells in an effort to make more powerful cellular therapies for blood cancers. So I think many people are familiar with CAR T-cell therapy, in which we can engineer receptors called chimeric antigen receptors. I'm showing a diagram of a CAR right here. And this is one of the most famous CARs, the anti-CD19 CAR, which is very effective in a number of blood cancers. So we can engineer receptors such as the anti-CD19 CAR, and then transgenically modify human T cells from those battling cancer so that they express these CARs, and then put the CAR T cells, as they're called, back into the patient. And once these CAR T cells are placed back into the patient, they show really strong responses, sometimes very long-lasting, durable responses. But there's a lot of effort now to improve the ability of these to kill cancer cells and also to last for long periods of time and make sure that the cancer doesn't return. So we want to be able to increase properties like what we call cytotoxicity, that killing effect, and increase stemness, the ability of these cells to maintain a long-lived stem-like state. And so in my work, what I've done is take small pieces of the signaling part of this receptor and broken them down into what's called signaling motifs, and then recombine those signaling motifs to make new combinations of signaling domains. And these give the CARs, and therefore the CAR T cells, different signaling properties and different anti-cancer phenotypes. So in my work, we test hundreds of combinations of signaling motifs, and then we ask, how do they affect the cytotoxicity and stemness of the CAR T cells? We can then take what we learned from these hundreds of results and use neural networks to predict the cytotoxicity and the stemness that will result from new and untested combinations of signaling motifs, and hopefully guide us to the best combination that's optimal for the type of cancer that we're working with. So here on the bottom row, I'm showing examples of cytotoxicity and stemness from nearly 250 different examples of CARs with modified signaling domains. Many of them have optimal or very high cytotoxicity and very high stemness. Some of them are even better at curing leukemia than the standard CD28 or 401BB co-stimulatory domains that are found on the market CARs. So we can generate a wide variety of anti-cancer functions, but then we can use neural networks and machine learning to pull out design rules. For example, we can learn the effects of placing signaling motifs at different positions in the CAR, for example, close to the membrane at this position or far from the membrane at this position. And we can see that this position close to the membrane, if we put a certain motif there, it may not be good at promoting killing of the cancer, but if we put it at this position, it's in fact very good at promoting killing of the cancer. And we can take design rules like the ones that we learn here, and we can apply them to design improved versions of existing CARs. So as an example, we can take this motif M1, which binds a protein called PLC gamma 1, and add 2 copies of M1 to the furthest part of the receptor away from the membrane. And we can see that in mice, this is better at controlling tumor growth than the standard co-stimulatory domain, which is found in CARs that are on the market. And so, I'd like to apply this kind of research to further develop CARs and other receptors that are really helpful in immunotherapy contexts.
Great. Thanks so much. We're going to go through the presentations. I know people will have questions for Kyle, but we're just going to go through the first 3 presentations briefly and then open it up for discussion. So thank you. Next, I was going to ask Allison Didychuk, who's to present maybe 1 slide on her work or 2 slides on her work. Thank you.
Great.
Hi, everyone. Yeah, so I was, as Julie mentioned, trained as a molecular virologist, but my background is in structural biology. And so what my lab studies now is kind of the very basic biology of viruses that can cause cancer. And Damon Runyon has supported me throughout my postdoc work and now in my independent lab. And I think that really highlights their commitment both to direct translational work while also the kind of basic underlying biology. And so I'll just talk to you for a couple of minutes about the topic that we're interested in. We study these two viruses called Kaposi sarcoma-associated herpes virus, or KSHV, and Epstein-Barr virus, or EBV. And these are two herpes viruses that infect humans, and they can cause cancer, particularly in individuals that are immunocompromised. And so they're part of this larger class of human herpes viruses, which you may have heard of. These are incredibly ubiquitous viruses that cause a real variety of disease, including herpes simplex, which can cause cold sores, varicella zoster virus, which can cause chickenpox and shingles. But KSHV and EBV are in this class of ones that are oncogenic viruses. And more broadly, they're part of this kind of set of infectious diseases that can cause cancer directly, which is thought to be linked to about 10% of cancers. The primary virus that I study, KSHV, is linked to cancers in some regions of Africa. And that's because, unfortunately, with untreated AIDS, it can cause cancer in those particular immune suppressed populations. And so KSHV isn't the most common virus, but it is endemic in some regions. And so there is a pretty big therapeutic need for it. In contrast, EBV is a much more common virus. It's thought to be, more than 90% of people are infected with EBV by the time they're adults, but it can still cause cancers in some individuals, like Burkitt's lymphoma. And so herpes viruses all have this biphasic life cycle where they switch between latency and then periodically reemerge into the lytic cycle. And this is where they are producing new infectious viruses. So you can kind of imagine this is when you get the cold sore, when you get really stressed. And so all herpes viruses have that. And that makes them kind of hard to treat and ultimately hard to cure. So even though we've known about herpes for a very long time, there's no cure for any of them. And we are in need of new therapeutics for the ones that we have with us. So here I've shown two small molecule drugs that are used to treat herpes virus infections. And these have common properties in that they both are targeting a single viral protein, this virally encoded DNA polymerase that helps replicate its double-stranded DNA genome. And when you target only one protein, you can end up with resistance mutations. And so there is a pretty big need for new therapeutics that are targeting other essential parts of the virus life cycle. On top of that, even though vaccines are great, and there's a CMV, cytomegalovirus, herpes virus, mRNA vaccine, hopefully in the works. That's not true for all of the herpes viruses, and there are other considerations for vaccines that might mean that we need additional therapeutics. So my lab focuses, again, kind of on this basic biology. In particular, one of the principles that when you look at an infectious herpes virus capsid, in this, you can see this is kind of the glycoproteins and this tegument layer here. And inside this capsid is the viral double-stranded DNA genome that's been packaged to incredibly high pressures, like 10 times as much pressure as a bottle of champagne. And we're interested in how that happens. It turns out that the virus encodes some core replication machinery in order to produce new infectious viruses. I already mentioned that it encodes its own viral DNA polymerase, and it includes some other machinery as well. We're also interested in understanding how it hijacks cellular transcription factors, including RNA polymerase II, to transcribe a subset of its genes, and how its virally encoded motor allows for this double-stranded DNA to be packaged into a nascent capsid. And so my lab, which started in July, is interested in unpacking the packaging machinery by looking at the role of accessory factors and figuring out exactly what they're doing by trying to take a single molecule biophysics approach to understand what's actually happening, the physics behind this packaging motor, but also trying to use the power of cheap library synthesis to try to take a more broad approach and apply techniques like deep mutational scanning, where you can have a variant library containing all possible mutations in a given protein and put that in and assay things in a much faster way. So I'd be happy to answer questions about oncogenic viruses and about Damon Runyon's commitment to basic biology.
Great. Thanks so much, Allison. And our third presentation for today is from Rabi Upadhyay, who's was a physician scientist supported by Damon Runyon, and now is an instructor at NYU School of Medicine. And take it away.
Thank you so much. And thank you for the opportunity to speak with everyone here virtually in real time or on YouTube, wherever we're meeting you from. As mentioned, I am a cancer doctor. I'm interested mostly in lung cancer patients, but it's hard. Things don't work as well as we want them to do. And we need to fix that. We need to think outside the box. And that's why the opportunity to do this type of research is so valued from the Damon Runyon and to do all the types of research that we're discussing today. This is the slide I wanted to present today. Kyle already alluded to this. This is on the right side here. That's where I'll start. These are actual T cells, the CTL, cytotoxic T lymphocytes, that you see here attacking a cancer cell, labeled TC. That's what we want to happen. That's what we want to, as cancer doctors, as cancer patients, and the greater community. This is what we want to happen. It doesn't happen, but we've appreciated this process for a long time. You can see the citation for this is back in the 1980s. And there's a variety of approaches, and Kyle mentioned some, and Allison mentioned some, as to how to engage the immune system to make the right side happen. And the thing that I'm excited about, that I hope to talk to you with everyone about today, is the concept of the gut microbiota. It's a fraught topic because it has already entered the lay dictionary through Oprah and all these other people who are promoting the gut microbiome as a cure-all for everything. And certainly one has to be careful as to what's real and what's not, but there is a lot of impactful things to be learned. Here on the left is this beautiful image from the Justin Sonnenberg's lab. And just to walk you through it, the blue cells on the bottom right of this image, that's actually you. This is our colon itself. So these are the cells of the surface of the colon. And some of these cells are actually secreting this green protein here, and that's actually mucus, that's MUC2. And so this mucus is being secreted by some of these blue cells, and it's creating these layers of mucus that coat the inside of our colon. And then beyond that, you see this beautiful community of bacteria that are labeled with various colors using techniques from the Sonnenberg lab, and we're doing the same in our lab. And you can see even just based on the shapes, the sheer scale of diversity of this universe of bacteria. And there's a lot of interesting things that people say, that people that study this, mostly to do with the amount. And if you had to put a number on it, it's around 3 pounds of bacteria that we all harbor in our guts, that we walk around with. But what we are very interested in is actually the genetic functionality. There's around 20,000 genes in us. And there's at least 10 times more genes in this universe of 3 pounds of bacteria that we carry in our gut. And even more than that, the genes in this pool, in this meta-organism, are being passed around. The organisms themselves come in and out through our lifetimes, from when we're born, when we travel, from our moms and from our households, and the bacteria interact. And there's a very exciting set of literature that shows that when you alter the gut microbiota, you alter your host immune system. You alter not only the cells here shown in blue, but the greater host, the whole mammal, the whole organism. And in fact, when you alter it, you can alter immunologic processes, again, like the one I show here on the right. And we now know that when you do large-scale changes, such as fecal transplant, which is now approved since November for C. diff and other conditions, when you give broad spectrum antibiotics, that has a tremendous effect on the outcome of cancer immunotherapy. And so because we see that strong effect, we now know that some of the genes in some of these bacteria are crucial for some people to have a response and others to not. And so as we study what is it that these gut microbiota are doing to the host immune system, what signals are they relaying, we can find the needle in the haystack. What is the signal that's coming from this microenvironment, making its all the way to the lung tumor or whatever tumor that you're studying, and having an effect and actually engaging this response that we want to see. And so there's a lot of work to be done, and we're setting up. I work currently in Dan Littman's lab, who's been studying this type of biology for a long time, and I think Julie mentioned she's been studying this as well, and many others. What are the mechanisms? What are the rules that how we interact with this ecosystem, with this universe of organisms? And I'll leave it there.
Great. Thank you so much, Kyle, Allison, Rabi. And I guess I want to start our discussion even by reflecting on how the three presentations, where are there areas for interaction there? And because I think some of science right now is about how we're seeing, life at the intersection between these fields. And so maybe with CAR T cells, is there some way in which you think, are we examining when you do cancer immunotherapy? Are we also thinking about how the gut microbiome is an important part of that? And how does that, how might that potentially impact the responses we're getting with CAR T cells? And then even to pull in Allison, there's going to be something about the history of our immune systems having to do with prior viral integration. And I guess in that context, it's like, I see all of you doing really complex scientific problems, but yet part of the real complexity is that we're trying to bring different fields together and then asking questions. So are there ways in which you're borrowing ideas and using ideas from other fields? And which of them are exciting to you and are you prioritizing? And I'm just going to pick on anyone who seems to be nodding their head to Kyle. Take us
Julie,
away.
actually, I have a slide I'd love to share that
Great.
I think addresses a lot of what you're talking about. So this slide, I think, addresses two ideas. One is more specific, and it deals with, for example, can we think about the gut microbiome when we're thinking about, engineering CAR T cell immunotherapy? But then there's the broader question that you asked, which is how do we bring together sort of disparate areas of cancer research and combine them to get somewhere that we might not get with any one of the areas? And, I titled this slide the way I did because I think there's a lot of wonderful research going on that's led to and is currently leading to improved cancer outcomes. But when I read the literature, I think the most striking and impressive results are coming from combinations of either new technologies or new technologies and old technologies. And so in this graphic on the left, I give the example of three technologies which in and of themselves are very impressive. The first one is the chimeric antigen receptor, which I spoke about earlier. The second is another receptor called SynNotch, which can recognize an antigen or a molecule on the surface of a cancer cell or another cell near the tumor and then activate transcription of a gene. And the third is what's generally called an armored T cell. And that's a T cell that's been engineered to secrete a cytokine, and that cytokine boosts the immune response. So all three of these are really powerful technologies. But recently there's work that's come out of actually the lab that I'm in, but it's work where these three technologies are packaged into one T cell, and that T cell shows remarkable improvement in its ability to clear tumor. So on the right, I'm showing, what it looks like when these technologies are in the same cell. So the CAR recognizes the cancer, the synapse receptor recognizes a molecule in the vicinity of the cancer to let the T cell know that it's near where it needs to do its work. And then that activates IL-2, which supercharges the T cell response. And you can see that if you just have the CAR T cell without this IL-2 production, it doesn't do a great job of clearing the tumor. But if you add this IL-2 production circuit, you get this massive reduction in tumor volume. So this is a much better anti-tumor T cell. And it's because it packages all three of these technologies and takes advantage of their ability to work together. And this also reduces, systemic toxicities. So it's potentially going to be safer for a patient. And I think there are other examples of combining technologies, not just in the context of CAR T cells, but in many contexts. So, I think about CAR T cells that overexpress transcription factors like c-Jun. So this is two technologies in one, and that's a better CAR T cell than what many of us are familiar with. I think there's a lot of really great emerging work looking at combining radiation therapy with checkpoint inhibitors. So this is a combination of old school radiation and new school immunotherapy. And then third, I think really bringing us back to one of the things you asked directly about is, can we think about the microbiome and its effect on cancer treatment in the context of CAR T cells? And Melody Smith, who is another Damon Runyon funded scientist, a lot of her work is in using diagnostics of the microbiome to predict immunotherapy efficacy, and I imagine to sort of predict what we should do to optimize and improve the efficacy of a CAR T cell therapy given the microbiome of a patient. So I think there's a lot of promise in these areas being very synergistic and leading to improved anti-tumor effects in ways that none of the individual technologies can do.
Kyle, I was going to mention the exact same thing. I was thinking exactly of Melody Smith's research. Not only that, in terms of efficacy of bone marrow transplant and CAR T cells, but multiple labs now have shown, Marcel Vanderbrink and others, that the gut microbiota actually affect the efficacy of bone marrow transplant and CAR T cells, the toxicity side, and I want to come back to that as well as a common thread between all the things that we're all studying, and basically how it tunes the immune system in various ways. One of the things to feed off of this, well, there's a couple of things to say. Julia asked, what is something that when we examine more of these things, there's a greater benefit. Part of the difficulty with all these problems is that they're incredibly complex, and you need to know where reality is, and you need more real data points to know where to set your reality. So when we start to examine some of these problems from different angles, I think that is very synergistic. For instance, when you try to figure out how the gut microbiota affect anti-PD-1 efficacy, you need to learn a lot of things, but you can also learn from, well, what are the gut microbiota doing to graft versus host disease or bone marrow transplant? What are they doing when you're having a viral infection, such as what Allison is interested in? And that also became relevant during COVID, as we all remember. The second thing I would say is sequencing, I think, was very dramatic for all our fields, right? Sequencing of viruses, sequencing of CAR T cells that were put in and what remains, and sequencing of the gut microbiota itself. That has enabled all sorts of fields in different ways to move forward dramatically. And to come back to one of the points I just mentioned, I think one of the common threads is very basic immunology, and knowing how is the host programmed to have tolerance and to avoid autoimmunity. We have a lot to learn from autoimmunity. And I'm going to make a point there to say that there's a lot of groups that already have made big headway into showing that gut microbiota affects autoimmunity. There are companies already that are developing treatments for childhood asthma. We appreciate that the exposure of kids to certain gut microbiota in their environments predisposes them to asthma or not, and that, frankly, that field is ahead of the cancer field, and they've been able to make more headway. But for all of ours, in terms of cytokine release syndrome for CAR T cells and obviously viruses, we need to understand how things work. We need to break them, and then we need to make models of cancer to kind of put it all together.
And Allison, what about in your field, this concept of viewing immune responses over a lifespan in the context of viral disease, and how that might be impacting effects months later, years later, but the ways in which our immune system is recording, our immune system is recording these microbial exposures. And then are we going to see, I mean, some of these fields, it does get complicated because you don't know, you can see an effect, but you don't know what actually caused this that you're seeing. And you think about it, that it would be interesting to be able to rewind the tape and find out what are the impacts here. But I suppose there is where we need to understand how to really understand large data sets, and how to find the real signal in the noise there. So in what ways are you seeing, I mean, it's a fascinating time, because of course everyone now has a much greater appreciation and understanding of virology, and we're just going through this massive rethinking of how a viral infection can have short-term and long-term sequelae. And it will be we're really changing a lot, because we've had so many people infected at the same time with the same virus. We'll now start to see, are we going to find a change in juvenile diabetes, or, these kinds of the work that we've seen, that now we're going to change it. But how is something like that changing, how we're seeing virology and its impact across other fields?
Yeah, I think that's a good question. And I'll preface this by saying I'm pretty far from immunology, but I think perhaps a nice example is the study last year of linking Epstein-Barr virus to multiple sclerosis. And so, these herpes viruses,
I just have to stop you, because, there are those papers that you hear about on the news or something, and you think, I'm going to go read that paper. And that paper was one of those for me that I just stopped what I was doing to read it, because it's something where it was an amazing study that you think it really did require that amount of data and that amount of setting up that study. But wow, was it big, the impact of both the work and the viral infection. So, just that I thought was, for many of these papers, that was for me a real landmark study showing the relationship between EBV and multiple sclerosis, and the fact that if you retain these kinds of data sets, you can find that was really impressive. What you'll say more of, but that it was a military recruit study, and that it was really impactful to have that many people who were the same age and then be able to track over time.
Yeah, absolutely. I think it was a field-changing study for sure. But I think it, as you alluded to, opens up the question of how did these viruses that we are constantly living with, whether we've been infected very young, like with herpes viruses, or we've been infected more recently with SARS-CoV-2, how is that changing? Yeah, as you mentioned, either periodically reactivating in the case of herpes viruses, or perhaps having more short-term effects in the case of, medium to long COVID. So this is definitely not my area of expertise. So, but I'd be happy to talk more generally about viral infections.
I think it's very relevant to even current public policy, because we're struggling with, people have probably seen in the news, there's an idea that we should now all be influenza style vaccinated once a year against COVID. But that, coming back to a basic science question, how much does the order of the vaccines that you got affect your immunity against that virus? Is it a good thing to get continually immunized against the same antigen? Is it a better thing to get immunized against different parts of that antigen? And then all those questions now relate to cancer as well, right? So if we're going to try to program ways to get the immune system to go after our cancers, what kinds of antigens should we use? How should we present them? These are all very basic and important questions. And then to the EBV point, I also found that incredibly fascinating. Whether they're causal, whether the immunity against the Epstein-Barr virus is playing a causative role in multiple sclerosis, we don't quite know. It's very provocative. The same thing can be done when you, I think Mark Davis's group, when you look deep into the T cells that are within lung tumors. I think he's observed similar epitopes, EBV and adenovirus, I believe. Again, I don't think anyone knows, are they just bystanders? Are they just hanging around and it's an inflamed environment and you're attacking the cancer, so now you've reactivated and you've infiltrated these T cells against these antigens that you were exposed to when you were a kid? Or maybe that's a big driver of how you actually get your therapeutic response. So maybe we should be paying a lot of attention to the colds and EBV that we got as a kid, and those are the cells that are doing the job when we have cancer in our bodies. I think they're all fascinating questions, but they're very difficult to answer.
So there's a question from online that's similar, and I've got a few questions from online. So one of the questions is, are there good viruses in the same way that there are good bacteria? We do talk about that a lot in terms of bacteria being commensal or beneficial. And a lot of what those bacteria are doing is taking up space so that you don't get colonized with a bad bacteria, something that maybe would be acquired in a hospital or something. And we talk about that concept of, good bacteria, basically that from an ecologic point of view, it's probably easier to create a symbiotic relationship with bacteria that are either neutral or beneficial than it would be to try to maintain completely sterile environments that you would constantly have them be exposed to microbes. So maybe it's just easier for the human body to not try to maintain sterility, but actually co-opt bacteria that are beneficial in terms of the gut microbiome, or even other surfaces that maybe they're even not providing that much value, but they take up space so nothing more pathogenic can invade. But in terms of viruses, coming back to this, there certainly has been some work suggesting that viruses sort of tickle the immune system. And maybe in that context, sort of putting it out there as, do you see a role for microbes to basically just make sure that the immune system is constantly doing surveillance, and that you wouldn't want in the same way that I'm saying, like, the human body, it's more energy to keep us sterile. Is it that there also is a role for microbes that you don't want the immune system to kind of get, fall asleep on the job or something? And or is it that these viruses actually do portend something that maybe you do need to draw the immune system to those areas? So kind of a broad question of just where are we in sort of, where are any of you in thinking about sort of host microbe interactions? Are there beneficial viruses? And if they are, what would those beneficial viruses do? Are there beneficial bacteria? What would they do?
I don't know if there's I can't think of a paper where there's beneficial viruses. As you said, there's certainly beneficial active bacteria, commensals that we see that we need to have. Certainly, I think a lot of us work with mouse models to study our projects. Certainly, when you take a mouse and you grow it up in a bubble, so-called germ-free mice, they're very deranged. If you look at their lymph nodes, if you look at their immune system in general, they do not develop the way they're supposed to develop. So now if you take a mouse, a mammal, or a human later on, and you look at its gut, you certainly find a lot of microbes, commensal microbes, and mostly bacteria. You certainly find fungi. I don't know if you see active viruses, but that's not, who's to say that potential viral exposures that we were exposed to gave us something beneficial? I think that's a hard thing to pin down. And the second thing that your question made me think of is, we mentioned sequencing. When we deep sequence our genomes, we see many remnants of endogenous retroviruses. And so I don't know how much of the audience is familiar. So these are viruses that we were exposed to that now became literally a part of our genome. And then again, coming back to cancer, we know that some of these instances of cancer immunotherapy work because they de-repress, they open up that part of the genome that's kind of been silenced. Our bodies have, we had an infection with the virus became part of us, and now we kind of cover that up and hide it with epigenetics and other mechanisms. But now if we have cancer cells and we either are able to, or spontaneously those viral elements are exposed, our bodies are hardwired to attack. So in the therapeutic setting, that's very beneficial. So I can't say, I don't know, I'm curious what other people think, but I can't say that active viral infection has been shown to be beneficial, but I certainly I wouldn't rule out that multiple viral infections and exposures might be beneficial for present times. I don't know, I'm curious what people think.
Yeah, that's also my understanding of it. I certainly can't think of an example of a virus that is good for mammals, at least. There might be some weird, plant virus out there that is beneficial, but certainly not for us. I think if there are examples, they're probably, infection with this virus makes you slightly less likely to be infected with another virus, which is kind of Robbie was alluding to. I think the way I very simplistically think of it is, viruses are, they're parasites, right? They're not bringing a lot to the table. They don't have a lot of genes. Bacteria have other things going on. They can make cool metabolites for you. They can help you process things differently. And that's just not generally true for viruses. Even the large viruses like herpes viruses, maybe they have 100 genes, but most of them are going to just, be for core replication things or modulating the immune system, but they're not bringing a lot to the table that might be potentially.
But you acknowledge that we are partly virus, right?
Oh yeah, for sure.
Maybe
over
ancient viruses. I showed that image of we can visualize the bacteria in our gut. They are part of us. But on a more conceptual level, the viruses are also part of us. They're just literally part of our blueprint at this point. So
yeah, absolutely. Over hundreds of millions of years, maybe signs or lines or whatever other, history of that is what is, helping shape our transcriptome in a different way. But I don't think an actively circulating virus, I think there's very few examples.
Yeah. So I want to bring it back to Kyle for a minute, because you came up with a model in the beginning. We got a question from online about, will combination immunotherapies be affordable? And in what ways, when we think about these breakthroughs, is there some concept of how much of this is going to be highly personalized, and how much of this is going to be entering mainstream medical care in a way that is affordable? Although it is pretty remarkable if you can imagine giving someone back 20 years of life, that you can make an economic model for them. But what are the, how are people considering those kinds of decisions?
Right. Yeah, that's a great question with a lot to consider. And I think, so first, I think there's there's a lot of effort now to think of ways to bring down the cost of of immunotherapies. So how do you make the licensing of the technology itself more affordable? How do you make the care that someone being treated with immunotherapy, how do you make sure that that care is more affordable? So if someone being treated can be out of the hospital safely in 2 days, that's much more affordable than someone who has to be monitored for 2 weeks to make sure that there are no side effects, right?
Yeah.
And then I think also there's some technological hurdles that, again, think about layering new technologies on, and we're answering the question now of, is that going to be affordable? But I think some of the technologies that are emerging may actually be what's necessary for this to be affordable. So if we think about things like allogeneic, T-cell transplants, so we're thinking of technologies where you can have sort of almost factory-made T-cells that are sitting around pre-engineered, and you can do some diagnostics on the patient and sort of pull out the personalized product that person needs, but because it's mass produced and in a way that's more efficient, perhaps that technology is much cheaper than having to make a bespoke treatment from the patient's own cells. And then, of course, I think, another hurdle that we have to overcome to make some of these technologies affordable and feasible is things like, can we get the genes that code for these many different technologies, can we get them into a T cell or another type of immune cell? And I think even the problem that Allison is working on is extremely relevant to this problem. How can we package DNA in a meaningful, efficient way? And currently we deliver a lot of DNA using lentiviruses. So, viruses may not be obviously helpful inside of us, but they're really great research and therapeutic tools. So we use a lot of lentivirus to deliver genes. But you could imagine, since herpes virus has much larger genome and it's packaged much more efficiently, if we can sort of take advantage of what Allison is learning, that would be a huge breakthrough, which itself might bring down the cost of these kinds of technologies in the future.
Yeah, that's
I echo
super
the
interesting.
I think the word I use for it is off the shelf CAR T cells. I think that's tremendously affects the cost. I'm speaking to you from a hotel room, and the venerable Carl June is at this conference, and he spoke about off-the-shelf T cells, CAR T cells as well. I don't know how much our audience is aware, but the reason that's so impactful for cost is because this is a very high, the stuff that Kyle does, I'm sure, is a very, very advanced technicality. And then to do it on someone's autologous blood, you need a very sophisticated on-site, it's called a GMP, Good Manufacturing Practice facility, to take a patient, withdraw their blood, infect them with viruses, such as Allison is working on, to encode certain types of receptors like CAR T cell receptors, and now give them back. And so it's like a very, Kyle mentioned the word bespoke, but that whole process, takes time. It's a lot of time to turn around so somebody walks in the door and how to actually generate the product. And it takes a huge facility on site. So this idea of, I think people in our audience probably know that when you donate blood, you have the type A blood, type B blood, whatnot. You can only give blood between certain types of people. The same is true for white blood cells. So we can't just throw around CAR T cells. And part of the reason for that is the molecules that are on the surface that you would attack the other person's cells. So we either carefully give you back your own cells, or we figure out the compatibility of these molecules on the surface. But as we try to understand this more, there probably are, and people have started to study this, Kyle probably can speak to this more, off-the-shelf CAR T cells where we can have T cells that don't have the molecules that would interfere with the way that our own immune system is programmed, but do have the apparatus to accomplish what we want. And that's a huge economic hurdle. And if whoever figures that out will bring down the cost tremendously, I think.
And actually, we're trying to do the same thing in the microbiome field of, there have been these attempts by companies to do fecal transplants. We don't understand what parts of the microbiome is beneficial. And so there have been trials to do, maybe these 4 species, and they haven't worked that well. But really what they're doing now is exactly that, with a GMP facility, making these complex consortium, but each microbe is is its own capsule.
Right.
And then we would figure out, oh, this person needs these 6 microbes, and this other person needs these 6 microbes.
Well, I think it comes down to, and this is kind of where my work in Dan Littman's lab is, we have mouse models, we have reductionist models where we can put in one bacteria that gives us a huge anti-tumor phenotype, and another very similar bacteria that basically gives us nothing. So there's something very fundamental in the genes of those 2 bacteria that we need to figure out. But even if you found out this quote unquote magic bug and the magic functionality, kind of to your point, you can't just stick a bacteria into a human. There's a huge ecosystem it needs to colonize. So there's molecules and there's metabolism. So maybe you need that magic bacteria, and then you need 4 other bacteria to support the engraftment of that bacteria into your gut microbiota. So you need nutrition, you need metabolism, you need to make space for that organism to survive. So there's a lot of considerations. But yeah, I think all this type of research, as long as once we learn much more about it, instead of doing things like fecal transplant, which it's like a sledgehammer, we don't know what's working, but something's working, we can come down to very mechanistic understanding and have it be targeted.
Yeah, I have to say, I think that's one of the, well, I'll ask you all what you think of as sort of one of the breakthroughs, but for me, it's also this shift that we actually pay respect to how are we going to make these treatments affordable, and really create medical breakthroughs that serve society. And it does, it is part of the discovery ecosystem where we do have to spend a lot of money to really understand the basic principles, but then figuring out how do we deliver this at cost to really achieve the maximum benefit for the maximum number of people. That for me is something which I feel like 5 years ago I didn't hear people talking about, but I feel as though I hear a lot more of respect for how are we going to deliver this kind of care at cost? And realizing that that's often part of the decision of what breakthroughs reach the patients. So wondering from you guys, from our discussion, are there technologies that you're really excited about and using? You guys are really all at the cutting edge. So we've heard a lot in the background about, sequencing and CRISPR and screens. And are there things, we'll just go around and maybe take a minute each. And, Kyle, what are some of the things that you're looking forward to in the next few years?
Are you speaking specifically about like bringing costs down, or just
no, in terms of just,
yeah.
What's
on
the horizon? What are you interested in?
Right. Yeah. So my lab actually opens at Stanford this spring, and so I've been writing a lot of proposals about the things I'm interested in. I'm particularly interested in sort of developing platform technologies that are what I call plug and play, and I think in biology and in technology, sort of the most impactful things have been plug and play. So, the ribosome has codons, and those bring in tRNAs, and that's a plug and play system. CRISPR, both biologically and as a technology, takes advantage of the fact that you can plug and play with different guide RNAs, and that changes your output. And so I've been thinking about how do we develop various types of receptors for immune cells. So these would be receptors like CARs, but also other sort of immune-related receptors and synthetic cytokine receptors that could control things like growth and survival of immune cells. And so what I think of is, these are very modular things that we can break down into components. And if we had predictive models of how the components work together, then we could, in an abstract way, use these systems in a plug and play manner where you sort of combine different modules of genes to make your whole gene for a technology, and then you put that into a patient's cells or into sort of the off-the-shelf cells that we've been talking about. So I'm really interested and excited about what breakthroughs we're going to see from modularity, not only in building new technologies, but in combining technologies together.
Nice. I mean, that you made it sound like you were making it more simple, but in fact, you're making it very complex. But at least with written rules. And you gave us such a nice example of understanding about, where putting the different modules in the different places in the CAR T cell and how that can impact. And so, Allison, we've got like 2 minutes left. What are you looking forward to on the horizons?
Yeah, I mean, I think I've actually been a little bit influenced by Kyle's work. Kyle, we met many years ago, and everyone loves sequencing, but the ability to cheaply and easily synthesize huge libraries of things has really kind of affected how I'm thinking about my work and how we can do things a lot faster. So that's one aspect of it. And I guess a more basic biology thing that I'm excited about is, continued advancements in cryo-EM to look at very flexible protein complexes and complex machines. And so we can start to see, how these machines are actually really working by, looking at different steps. I'm excited by that, and by, cryo-ET, where we can start to see these things in an actual context. So this is you're taking a kind of a slice through a cell, and then now we're almost at the point now where we can start to see things in their real environments. So not with purified proteins, but how they're actually working inside a cell. And so I think the next decade will be really exciting about figuring out how all of these kind of core molecular machines, both in our cells and in viruses, how they're working.
Nice, yeah. And what about Rabi? You're going to take us out. What's on the horizon?
I think the words that when you were saying how to bring down costs, what really changes the game, scientists, we often use the phrase necessary and sufficient, right? So we have an experiment, we have a phenotype, we have something, we pipette something on top, and we get a phenotype, and it's amazing. And the question is, what is necessary and what is sufficient? And to define that is the most important thing, because that brings down costs, right? So we know CAR T cells work, but they're incredibly complicated, they're incredibly expensive, they're incredibly cumbersome. And we most likely do not need all the functionality. We need to know what is the bare minimum functionality, so we can make more of them, make them more fast, make them more quickly, and more with less expense. Same with bacteria. My pipe dream, as I said, we have these model organisms. One is efficacious, one is not. But really, to understand the genes that are at the level of these two bacteria, and once you understand that, then you can actually take a person's own microbiota, or take off the shelf Oprah Winfrey lactobacillus, the yogurt in your grocery store, and genetically program that to have the functionality that would be necessary. And I think that's the key thing, and that's why science is so hard, but that's why it can be so productive. Because if you obsess about the necessary and sufficient, you can win all those games.
Great. Well, thank you all. I've really enjoyed this discussion, and being able to meet you all and learn about your work and the future that you see. So I'm going to turn it back over to Young.
I just want to thank all of you for a really fascinating and thought-provoking conversation today. We appreciate you sharing all of your insights, and I think we can all be really optimistic about these new advances and progress that will lead to better understanding of cancer and improved treatments against the disease. So thank you all again, and thank you to the audience for joining us today. Please keep an eye out in your email for a link to complete a post-webinar survey and provide your feedback and suggestions to our team for future events, and stay tuned for emails announcing our upcoming Catalyst webinars later this spring. So thank you all again for joining us. We will have a link available to the recording of this session if you'd like to re-listen to any of the conversation or share it with others. Thank you again.
Thank you so much.
Bye.
Bye.